Yes, so in the last class, yes, we were discussing about the DNA is a genetic material, but many people were not satisfied with the answer, right? They were claiming that protein should also be the genetic material. Why? Because we all have the protein, and obviously it should pass into the next generation. So, of course, two scientists came to give their theory. So now we have doubt in which two organelle. Now people have doubt in which two organelle. Like they don't have any doubt in the lipid, they don't have any doubt in the carbohydrate. But yes, they were claiming that protein should be the genetic material, right? So right now we have doubt in what. I can't hear you. I guess you are not audible to me.
Oh yeah,
Yeah.
they ruled out RNA too, right?
Yeah. So actually, no, 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 not RNA. We can like they have a doubt in DNA and RNA. The
Oh,
theory
okay.
was claiming that DNA is a genetic material because of the Griffith experiment. But other people are
Hmm.
saying that, like those are not so educated, they were saying that protein should be the genetic material. Why? Because it is the we can see it by our eyes. So how can we say that? So that's
Hmm.
why now I am going to a another experiment, and that experiment was given by Hershey and Chase. Hershey
Hmm.
and Chase. So these two scientists try to work on protein and see, it was completely clear DNA is a genetic material. But for clearing out the doubt, so they performed the experiment to differentiate between the protein and the DNA. So Hershey and Chase, do you know what do you mean by the radioactivity?
Yeah,
So what do you mean by the radioactivity?
em- emitting radiation.
Yes. So actually you can make anything radioactive by taking the radioactive substance, right? So actually,
Yeah,
okay, if I ask you one thing, can you tell me what is a bacteriophage?
no, I don't know what that is.
Okay, bacteriophage is a virus that attack on the bacteria. Okay,
Oh, okay.
bacteriophage is a virus which attack on the bacteria only. So it actually it transfer it is replicating, it is reproducing with the help of bacteria. So the proof of DNA is a genetic material. It is done by the Alfred Hershey. Proper name is the Alfred Hershey and the Martha Chase. Alfred
Oh,
Alfred Hershey, Alfred Hershey and Martha Chase. Okay.
a woman
So
for once.
okay, so they performed experiment, and it is the last experiment that will definitely give you the idea about finally DNA is a genetic material. So the bacteriophage is a virus that attack on the bacteria. So can you just see that? Something like
Yeah.
this.
Mm-hmm.
It's not a spider.
I
It
know
is
it's a
just
virus.
yeah. So this is virus, okay? And see, whatever you can see inside, this is actually the genetic material, DNA. And whatever you are seeing the outside, the tail, neck, the protein capsid, it is what? It is the made up of protein. Okay. And DNA, I am making it by the blue color.
Mm.
Okay. Now we have one more bacteriophage. Oh, sorry. One second.
Wait, how are you like drawing this? What, like, are you using a whiteboard? Zoom whiteboard?
I have a pen tab.
No, like I know, but like, where are you like drawing this?
Yeah, yeah, yeah. It's whiteboard. Yes, yes.
Oh, I can't hear you. What? Hello?
Yeah,
Okay. Hello?
yeah.
Okay, there we go. I see. Okay.
Yeah. Okay. So as I told you, this is what? This is the DNA, right?
DNA.
Yeah, genetic
Yeah.
material. It could be RNA also, but this is the main DNA. And Yes, this is the main protein as I already showed you there. I know you are thinking why I did why like why there are two diagrams, right? So
Yeah, why are there
actually I'm
two?
see because of the radioactivity,
Oh,
because
it
of the
multiplies.
yes. See now what I'm doing.
Okay.
I have made this protein radioactive. See very
Oh.
carefully. So now yellow color I am making just to show you the radioactivity. So just tell me yellow color you will see only in the protein or in the genetic material also.
Uh,
See.
in both, and only the protein,
Yes, and if I show you like this, here radioactivity is where?
the DNA.
Yes, it means we have made the protein of the bacteriophage radioactive here, and we have made the radioactive DNA here. Okay,
Hmm.
so we have made the DNA of this bacteriophage by using the phosphate, I mean phosphorus group, and we have made the radioactive with the help of the sulfur. Sulfur is also radioactive some part, and because of the phosphorus, we have made the DNA radioactive. Now, of course, now we have what? We have the bacteria. Actually, this bacteriophage transfer the material for the reproduction. This is the bacteria. So if this is the bacteria, so of course now what this bacteriophage is doing, it is going to infect. We allow this bacteriophage to infect on this bacteria. So this is the infection state. We are doing this thing in the test tube. Okay, we have put like so many bacteriophage in that. I mean the radioactive one, radioactive protein and radioactive DNA here. So now what will happen? We have allowed the infection state, then we are completely mixing it, and then we are doing the centrifugation. See, we are blending means we are mixing it, and then we are doing the centrifugation. Centrifugation means the lighter one will come above on the test tube, and the heavier will remain at the downward side in the test tube. Okay, so you know it very well, the bacteriophage are very light, so they will remain above, and the bacteria are heavier, so they will remain downward like this.
Wait, so say it again.
Yeah, sure, why not? See, we are doing all this process in the test tube.
Oh
Bacteriophage
no,
is
I got
very
that. But what are the three like terms that you used?
three like okay. Infection means we are allowing this bacteriophage to infect on this bacteria. It is landing
Hmm.
on that. Blending means we are mixing it,
Yeah, infection,
blending.
blending, and centrifugation.
Centrifugation, and now we are separating
Centrifugation.
it. See, why we are separating it? Actually bacteriophage is quite lighter as compared to bacteria is heavier, and this bacteriophage is lighter. So on the above side you will see heavier one or lighter one.
On the downside, you're gonna see the heavier one
Yes, heavier
because
it
it
means
sinks. It
bacteria.
sinks. It sinks.
Yes, and it is lighter one. So now when we allow the infection, so see very carefully, Vishnu, very carefully.
Yeah, I see. Yeah.
Now in this state we didn't see any radioactivity in the bacteria. No radioactivity came in the bacteria. No radioactivity in bacteria. But in this case, radioactivity have been observed. There was radioactivity in the bacteria. Now, from this thing, what can you conclude?
DNA is genetic material.
When it tried to infect this, so it has transferred the protein or it has transferred the DNA.
Transfer the protein,
uh if it has transferred the protein, so it would become the radioactive.
but it's not. So it's just DNA.
Yeah, so it means it has transferred the DNA and there was no radioactivity. It means because people were saying protein is a genetic material, so if
Okay, so
protein.
okay, I think I know. I think I know what you mean. If the DNA was infected, then it would make another thing, another virus, right?
Yes, exactly.
Oh, I
See,
see.
here DNA was radioactive, And
And
now
now it
we,
makes another thing. It makes another
yeah,
virus,
so it means
bacteriophage.
it has transferred the DNA, right? That's why this bacteria also become radioactive,
Yeah.
isn't it? So
Yep.
like this. So it is something like, well, like suppose if I am going somewhere and I am just dropping a white color everywhere. Okay. So if there is a school and if people are saying I was there. So if I was there, so obviously you will see the red color.
Hmm.
Are you getting what I'm saying?
Yeah.
Suppose if I'm going anywhere, so I am just dropping some red color. So but some people are claiming that you were there in that school, but in that school there is no red color. So I was there or not? No, I was not there. Okay, so same thing is happening here. Here protein was the radioactive, that's why it is not going to be transferred here, because protein is not a genetic material. If protein was the genetic material, then obviously you would see here.
Yeah.
Okay, so radioactive would have been observed in this bacteria, but it is not. So that's why, because of this, we can conclude that which thing is genetic material?
DNA.
DNA is genetic material, not the protein, because protein is not getting transferred. Okay, see, if protein is genetic material, so of course it should not show the radioactivity, because here protein was not radioactive, and but it is showing radioactivity. So it is very simple. Got it?
Okay, yeah, I got it. But what are the dots in the test tube up there? Is that the bacteriophage?
What is dark? What is that?
You see the test tube diagram like on the top of the page?
Okay. Now, so if I draw it
I hear you
here,
again. Okay.
so see, within the
Hold
test
on
tube.
1 second. I can't hear you. Okay, yeah, I can hear you now.
Actually, these things we are observing in the test tube. So at the above side, we are going to see the bacteriophage
Okay,
because they
that's
are
what I thought.
lighter.
That's what I thought.
So in the first case, bacteriophage protein was radioactive or DNA.
In the first case, protein was radioactive and nothing
So
happened.
it means radioactivity you will see above, and the
Yeah.
colonies of bacteriophage, it is called supernatant.
Hmm.
Colonies of bacteriophage, they are called supernatant. So supernatant were showing radioactivity, but protein were not showing radioactivity. It means they haven't transferred the genetic material, they haven't
Super
transferred the
natant.
bacteria. There is no radioactivity. If I talk about here, so these are the bacteriophage.
Oh, I see.
So
Because
here,
the bacteria is not radioactive, that proves that the protein is not genetic material.
yes, because they are not transferring that.
Okay, I
Yes,
see.
when anything is in your DNA, so obviously you are going to transfer in next generation. If whatever, like suppose if I have a DNA, but suppose something like, because like my protein structure is getting ruptured, my protein structure is getting like some problem, but it is not going to be transferred in the next generation, because protein is not a genetic material. And here radioactivity was transferred in the bacteria because of the transmission of DNA.
Because okay, I'm gonna label case one, case two. Okay, because case one.
So can we say that it is found that when the phase containing the radioactive DNA, bacteriophage containing the DNA, so it has infected the bacteria, it has transferred its DNA into the bacterial body, and it is indicating that the DNA was injected into the bacterial cell. So DNA was injected into the bacteria from the bacteriophage. That's why we can conclude bacteriophage. That's why
Okay, I think I understood this concept really well.
we can conclude,
I think I understood this concept pretty well.
okay, DNA is a genetic material.
Yep. I added like a little conclusion note to myself too.
You have some confusion.
No, I wrote a conclusion note on my paper.
Okay, 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 fine, fine. Okay, so this is. Oh, can you tell me what is a supernatant?
Supernatant is the colony of viruses that are
Yes,
not or yeah,
bacteriophage. Okay, so
bacteriophage.
finally, theory concluded DNA is genetic material. Now we don't need anything. Just please shut up, everybody. Those who are saying protein, 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 just go back to home. Protein is not a genetic material, and it is concluded that. Okay,
Yes.
so these people come back again, and they say, they ask actually, what is genetic material? Can you explain? You
Oh.
are saying DNA is a genetic material, fine, but genetic material should also have some property, na? Tell me what is genetic material.
Me?
So, no, no, those people I usually ask.
Okay,
But
okay,
yes,
okay.
you can tell me also, what do you understand by the genetic material now? Yes,
So
I'm
genetic
asking you.
material is the hereditary properties that goes from the parents and that's also transferred from the parents to the kids or the offspring.
Is it the only thing by which you can
Yes.
say genetic? Okay, but except this line, there are more thing. Okay, so
Oh yeah, yeah.
first of all, okay, DNA is genetic material, fine. So it will remain only one, or it keeps on dividing.
It keeps on replicating.
Yes,
Yes.
replicating, right? So can we say genetic material is something which should be able to generate its replica?
Oh.
Replica means its copy, replica. So genetic material means it should be able to generate its
Wait,
replica.
that's but cells can regenerate too. Stem cells can regenerate too.
So it can regenerate because
Oh wait, wait,
because
okay,
of
okay.
the
I can't hear
DNA.
you again. One sec, one sec. Okay, go on, go on. Continue.
Cells
Go ahead.
can regenerate, stem cells can regenerate because of the DNA. Without
Okay.
DNA, they can't regenerate. It is mainly the DNA.
Okay.
Primary, first thing is DNA, then protein, and then it is overall protein. You have seen that in the meiosis, mitosis.
Yeah.
DNA is the thing which is generating, and then chromosome formation, and then cell is going to divide.
Yes.
It should be okay. Your DNA should remain stable, or your DNA should combine with your food or anything, your body.
No.
It should remain stable, or it should be reactive.
Remain stable.
Yes. So it should be chemically, because if it is combining with your food, so then you won't have any DNA, because your all DNA is combining with your food, and now it has removed from the body. No, 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 no.
I'm not a person.
Yeah. So
I'm just a being
so chemically and structurally stable. So
with
it should be
genetic material.
chemically. These are the properties. Okay. Variations. Can you tell me what are the variation, Vishnu? Vishnu.
Yeah, hello.
Yeah. What is?
The new traits formed by offspring when they're born, right?
Can we say some changes like
Oh
my
yeah,
parent?
changes,
Some
changes.
slow changes. Slow
Yeah.
changes are good for a human body, right? For the evolution. Variations are the
Yes,
key for evolution.
yes,
That's why we are
yes,
going to evolve and like very
yes.
modify. So we should provide the scope for the slow changes. That is very good. So DNA should provide slow changes or not? That's why.
No.
No,
But
no,
yes,
no.
yes, 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 yes,
That's
yes,
why we are
yes.
we are becoming very advanced, right? Generation
Yeah.
after generation. So it should provide. These are the properties like for being like being a genetic material, it should offer these properties. Okay, it should provide the scope for slow changes. Okay, and why? Because for the evolution, obviously. Second, can we say it should express itself in the Mendelian character? Like, do you know we have discussed about the Mendelian cross, how
Yeah.
generation are going to express their character. So you can
Mm-hmm.
also write that it should be able to express itself as a Mendelian character. Like how much, what is the ratio, how much tallness factor, how much dwarfness factor, how the characters are going. So it should follow a Mendelian rule also, Mendelian character. Okay.
Yeah.
Okay, so it is about the genetic material. Okay, you should know one thing. From the DNA has a two strand like this. So using of one DNA, one strand, by using the strand of one DNA, actually RNA is also coming. This is known as
Oh,
mRNA. mRNA is single stranded. mRNA is single stranded, which is coming from the one strand of DNA. Okay. This is actually tRNA. tRNA is something look like this. And rRNA is something look like this, like a burger, something like this. So this is so tRNA are basically of three type: mRNA, tRNA, and rRNA.
how
Okay.
often do like DNA form? Like all the time?
Yeah, most of the time your cells are dividing. Yes, they take rest also. I told you in the synthetic phase, you remember? Interphase, G
But
naught phase,
is
interphase.
there
They,
a is there a way to like know where like right now where in my body like what time like
no,
where
no, no, it is not because you have so many
oh
cells. So in some cells, DNA is getting generated. Some cells are doing it very slow. Some cells are doing it fast. So it is like that. And some cells are going to die, and new cells are going to form. So it's like like your body is not going to take rest. It is
mm,
taking place continuously with some high speed, low speed. Okay, and at this age, it is actually fast process. Okay, because you
mm,
are quite young. But yes, after 35, after 40, it is going to be slow down.
mm.
Okay,
Does
so
DNA have any correlation with metabolism?
metabolism.
Yeah, does it have any correlation?
uh no, actually here, if you have the DNA, DNA actually help in the formation of mRNA. I will going to tell you this thing. So this is known as the transcription process.
Oh, yeah, yeah. Hold on, let's write that down.
From the
DNA.
mRNA, your body protein, which is coming, which is same like your parents. So this is known as translation. So if your DNA is quite active, so obviously it is going to form more and more protein in your body. So of course your DNA is linked with the formation like anabolism. Your
Yeah.
protein, your body protein, your face protein, your body, okay, the protein is accumulated here. So it is all because of the DNA.
Okay.
Because all the human being eat almost same kind of food, but still they have a
Mhm.
different protein structure. So it is all
Yeah,
because of the
that's
DNA.
so that's why some people can build muscle and like lose fat more than others,
Yeah, yeah,
faster than
yeah,
others can.
exactly. Okay, so now here, if I talk about the DNA and the mRNA, RNA is what? RNA is a ribonucleic acid. Ribonucleic acid, you know it very well. So ribonucleic acid contain the sugar, and this sugar is known as the ribo ribose, actually ribose. So this
Hmm.
sugar is known as the ribose, and if I talk about the deoxyribose, which is for the DNA, so
Mm-hmm.
deoxyribose. So ribose sugar and the deoxyribose sugar. So now it means for the RNA it contain ribose, for the DNA the sugar is deoxyribose. So what is the difference in them? Any guesses?
Hmm.
What is the difference? Can you predict the difference?
There's one less oxygen. O5.
I guess I told you this thing right in the biomolecule.
I don't remember. Maybe. Yeah.
Okay, I told you the ribose contain complete structure, but if I talk about the deoxyribose or deoxyribose at the carbon number 2, there is an absence of oxygen. So
Yeah.
that's why,
C6H12O5.
yeah. So now I am going to show you that structure so that you can see the main difference. Okay.
I got it right though. I got it right, right? Was I correct?
Yes, can you just see that? Yes, yes, this. At the right side.
Yeah.
See, at the carbon number 2, there is a OH. But at the carbon
Mm-hmm.
number 2, there is only one H. So
Yeah.
that's why there is a loss of one oxygen. Loss. So that's why it is a deoxyribose, loss of one oxygen. So you have to remember sometimes there is a question. Ribose is actually work as a catalyst. Catalyst.
Mm.
So why
It
ribose work as a
speeds up
catalyst?
the
Because
process.
yeah, actually it has a 2OH group. You have to give the reason. RNA has a 2OH group. That's why it is very reactive. It
Oh,
work as a
okay.
catalyst. But DNA is not so reactive because it doesn't have 2OH.
Our
Okay.
name
RNA
is
is working
is
as a catalyst because of the 2OH group. Okay.
because of the two OH group.
Yes.
OH
Group.
OH. Okay. That.
Got
Oh,
it now,
that's
this
good.
structure?
Yes.
Okay. So now, if I ask you how many types of the RNA we have,
Three.
can you tell me the name?
mRNA, tRNA, and RNA. rRNA.
Yes, so if we talk about the mRNA, so mRNA is known as messenger RNA. So actually it is
Yes.
coming out from the DNA. So can we say that it brings the information, it brings the genetic information from the DNA? Because whatever the information your DNA has, so it is being transferred to the RNA, and then this RNA help in the formation of the protein, that particular type of protein which your parents have, right? Similar kind of protein. So we can say it brings genetic information of DNA. Okay. And they are bringing the genetic information of DNA, and then finally it help in the protein synthesis, isn't it?
Yeah.
I told you, no, this process. See,
Yeah, I know. It's
mRNA is
translation transcription.
mRNA is bringing the information from the DNA, and then it is helping the formation of protein, which your body has.
Yes.
And mRNA is double stranded, single stranded.
Single.
Okay. If I talk about tRNA, tRNAs look like something like this, as I already showed you. Can you just
It
see
looks like
that?
a. It looks like the thing that like car
Christian.
mechanics use.
Yeah, a Christian symbol also.
Oh, the cross. Okay, okay, the
Yeah,
Holy
yeah, yeah,
Cross.
yeah, yeah. Are you Christian?
No,
Okay, okay,
my
okay.
name.
Yeah, your name, you're Hindu, right? Vishnu.
Yeah, of course.
Okay,
Yeah,
okay.
yeah,
Don't do like this, okay? It's like this. Do you know what is that?
yeah.
Like goddess
Sudarshan
Vishnu has
Chakra.
Sudarshan
Sudarshan
Chakra.
Chakra. Yeah.
Oh my God, you know it's Sudarshan Chakra. Okay, good.
Had some
Okay.
traumatic experiences with that.
Okay. Okay. So if I talk about the tRNA, so tRNAs, it act as a actually adapter molecule, and it see, I'm going to tell you the exact process of translation transcription, but later, not now. So as of now, I want to tell you it help in connection with the amino acid. Why? Because
Hmm.
for protein formation, you have to connect the amino acids,
Yes.
then protein will form. So this tRNA brings the
Peptide bond.
yes, so it brings the amino acid, okay, and it help in attaching them and then form the protein. So you will say,
Oh, after class, I have a couple of questions like regarding school homework, if that's okay to
oh,
ask you.
okay, okay. So adapter molecule that reads the code on the one hand. I
And
will tell you the code. Don't worry. Code means like those thing I told you: adenine, thymine, guanine, cytosine,
oh,
like
the imaginative
DNA.
bases
Yes, this is known as the code. every
code. All right.
every human has a different genetic code: ATGGCC, ATATGC, something like this. Okay.
Okay.
So one hand, so actually it read the code in the one hand, and it combined with the other hand and bind with amino acids,
Okay. I have a question.
binds to the एक second, bind to the
Yeah.
specific amino acid on the other hand. On the other hand, yes, say.
If I genetically make or like a genetically engineer a human, let's say if I have
No,
like eight.
it's not so easy, and they
Oh
tried well,
yeah.
but it is very difficult to make.
Okay, yeah. Can they reproduce?
Yeah, you can reproduce, but with
No, no. Like, can the person that I made out of a test tube
no,
can
no,
live
no,
on?
no, 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 no. If it is artificially, so most of the time we tried, but so whenever we are like we were trying for the formation of the artificial animals, and we found that they are sterile. They are
Yeah. Okay.
sterile. They
Okay.
are not able to like reproduce. They were not
Can you
able to
like?
reproduce. Okay.
Why not?
I don't know, maybe some genetic problem, some genetic complexity. Natural is natural. You can't
Hmm.
make, yeah, science is like doing some advancement, but we are not, we haven't reached yet actually that advancement level. So, yes, things are on the, you can say in the pipeline, but not like we can't make human,
Um,
artificial human.
I want to make my like. I want to make another version of myself.
Like yourself.
Yeah.
So there is a age difference. So how can you?
No, no. Like, I want to make him like so I can be in two places at the same time.
Okay,
You know?
come back in the real world.
Yeah, yeah.
Okay,
It's not gonna
okay,
happen,
it's
but it would be cool if it did.
okay, okay. I respect your imagination and I respect your project, but
It's
okay.
not gonna
So
happen.
yeah. It's yes, some practical thing and hypothetical things are you have to differentiate in between them. But yes, at
Hmm.
the science level, you can try like start from the beginning at least. At least if you're
Yeah.
thinking like in a big level, so start from the smaller one. Okay. Okay, so adapter molecule. See what I just told you. This thing help in reading the code, and this thing help in attaching the amino acid. And I will tell you how they help in attaching. But as of now, you have to remember this thing will read the code on mRNA, and this thing will help in attaching the amino acid. Can you just see that? So this tRNA molecule help in attachment there, and this thing help in reading the code.
Hmm.
Okay. And ribosomal RNA, I already told you, rRNA, ribosomal RNA. Actually,
Mushroom.
yes, burger-like structure,
Oh,
mushroom.
burger.
But okay, it seems like mushroom also. Okay, but actually, it is a burger-like structure, and it
Hmm.
plays a catalytic role. It help in the
Okay.
catalysis. Catalytic role actually it help in the process of like protein formation. So it has a catalytic role during the translation process from the mRNA to protein. Okay. Because you have already heard RNA is ribosomal, and ribosomes are very famous for the protein synthesis, isn't it?
Yeah. Yes,
So
sir.
rRNA means R means ribosomal, and ribosomes already you know it. Ribosomes help in the protein synthesis.
I
Okay,
like the
so
word synthesis. It sounds
yeah,
cool.
okay, okay. Now we are going to study about the replication of DNA. How DNA is going to be replicated in our body? How?
How?
How? You have to tell me.
Mitosis. No. No.
Mitosis. We are not discussing about the cell.
Transcription.
See. I'm telling you. See, this is your one DNA which you got from your parents. Okay. Now, what is happening? The one strand is going to be separated from this, like this.
Oh, it
And
unravels.
now, see, DNA is getting separated like this. Okay. And now, because of the, see, first you have to just say it, DNA helicase.
DNA helicase.
This DNA helicase is actually removing this helical structure and is trying to
Oh.
separating them. Now, what is happening? There is a DNA polymerase.
Wait, hold on, hold on, hold on, hold on. Let me write that down.
And I
Replicate.
will tell you, I will tell you. Don't worry. Don't try it right now. So DNA helicase is
Okay.
separating the DNA. Now
I know how to spell it.
DNA polymerase, the new DNA is coming over here.
Hello?
Hello.
Oh,
New
can you
DNA.
hear me?
Yes, I can hear you.
Okay, I can't hear you.
Now the new DNA is coming
Oh.
here. New DNA. So now initially it was only one DNA, am I right?
Mm-hmm.
Now first they just separated and the new DNA is coming like this. So now how many DNA we have?
Oh,
Now how many
I
DNA we have?
see. Three.
Three. No, from the one it has
Oh, two,
converted
two,
into
two,
this.
two, two.
Same like you
Okay.
did for the
That
cell.
yeah, that it's four, not six.
Okay.
I remember
Now,
that.
now they are further becoming large and now they are again separating.
Wait, what is the red structure again?
One second, one second. I'm going to tell one
Yeah.
second. So it is again
Mhm.
separating now. It is also separating now. So
DNA
now,
looks like a sine wave.
now the new DNA coming like this. So new
I have a
strand,
question.
DNA polymerase. I will tell you, don't worry.
Oh, that's polymer. Oh, I see.
DNA polymerase is coming over here like this. So now can you see how your DNA is going to be replicated from 1 to 2, 2 to 4, 4 to 8, 1, 2,
Oh,
2 to 4, and then from 4 to 8, 8
six,
to
eight to
16,
sixes.
like this and this, so
Oh,
forth and so on. Yes,
oh, it's an exponential function, right? One to the
yeah,
power of
yeah,
x.
yeah, 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 you can
Okay.
say that. Yeah, you was asking
Oh
something?
wait, that's okay. So is there like the? I noticed that the DNA, like the red and the green structures, they look like sine waves. Is there a reason why?
Different sign.
No, they look like a sine wave. The
Okay, okay, okay. Okay, just ignore that. Yeah, it is
oh,
like a
it's just for
sine wave.
drawing purposes.
Yeah,
Okay.
it is for drawing purpose and not a sine wave and cos. There is no physics here. Okay. Okay.
Okay.
So Watson and Crick had proposed this scheme. Okay, they have a double helical structure. So Watson, remember, Watson
Mm.
and Crick gave this structure, and this is the double
Yep.
helical structure. Watson, just say it. Watson and Crick.
Watson and Crick, James Crick, Francis Watson. Uh,
Mhm.
So is the green is DNA polymerase, right?
The green is a DNA which is polymerized by the DNA polymerase enzyme.
Okay.
We need a catalyst, and the catalyst name is DNA polymerase.
Which DNA, which is catalyzed by the DNA no
DNA helicase.
polymerized.
Polymerizes means polymerization, like from 1 to 2, 2 to 4. Like suppose if I have one hair and now I have millions
Uh,
of hairs, so it means my one hair is getting polymerized to form so many hairs.
Polymerase. Okay. How about the red?
Red is also for the polymerization.
Okay, yeah, yeah, okay. I'm just making
One
sure.
one is going to get polymerized into two, and then these
Mm-hmm.
two are polymerized into four, then these four are polymerized into the uh,
Yeah, 8. I'm just making sure that red and green are polymerized by the same enzyme.
yes, yes, yes.
Okay, but I'm DNA polymerase. That's an enzyme, right?
Yes,
Or protein?
don't DNA polymerase A's. Whenever you see A's, so it is always enzyme.
Enzyme. Okay, okay. DNA polymerase
And their
enzyme.
helical structure is removed by.
DNA helicase.
Hmm.
Okay.
But there is a one more enzyme. Its name is DNA ligase. I will tell you what they do actually. I will tell you later. Why? Because actually sometimes these strand is going to be broken. See, whenever they are going to form a copy now, so some DNA is like this. They are going to be broken, something like this. So these DNA has to be attached. See it here.
Yeah, I'm writing. I'm writing.
Sometimes DNA is broken in middle of these, something like this. So these structures have to be joined. So these structures are joined by the DNA ligase. Without DNA ligase, you can't join these structures. Okay. So these structures we need to attach, we need to join, and we can join it by the DNA ligase. DNA ligase. Okay.
Hmm.
So now, actually, there were two scientists, one more, last two scientists, actually, you can say. So their name were Matthew Michelson, Matthew Michelson, and Franklin Stahl.
Hmm.
Matthew Michelson. and Franklin is tall, and you can say Messelson is tall. Okay. So actually, they were explaining how how DNA is getting replicated, and they have a semi-conservative nature. See. If I am saying this is a black DNA, this is a black DNA, right?
Hmm.
So if you see this DNA, so this DNA has a new green strand and a one old black strand.
Hmm.
Suppose this black is your parent DNA. Parent DNA. Vishnu,
Yes,
listen
I see
carefully.
it. Yeah, yeah,
You
yeah.
you have your parents' DNA. So you
Hmm.
have something new strand also, yes or no?
Yes, because
New
it's
strand.
the.
See, if I consider that this black is a parental DNA completely, so when it is getting separated, so a new strand is coming here, red,
Yep.
new strand. Now, when they are going to separate, then you can see this again new strand is coming. So this is how variation is going to take place. So Frank Michelson and Stoll try to explain the same thing. See now very carefully, you have to see that. See, this is a one DNA. They took from the bacteria Escherichia coli, they
E.
taken
coli,
out
yeah.
E. coli, they taken out this bacterial DNA, and they saw that both with the help of, because they grew the bacteria into the NH4 chloride. I mean, you can say NH4Cl, like ammonium chloride. So that ammonium chloride was N15. So
Mm.
if you are putting this bacteria into the ammonium chloride, which is a N15, so of course their strand are heavier one. Their strand contain N15 and it is also N15. They are
Yeah.
heavier strand. Okay. Now, now, if you put this into the medium of ammonium chloride, but it is a 14 nitrogen, not the 15
Whoa.
one. So now of course with the help of the helicase enzyme, they are going to get separated now.
Yes.
They are going to separate, okay, and it is also going to get separate. So now new strand will come or not?
Yes.
So new strand will be, they like this strand is N15. Why? Because it is was N15 and this is also N15. Oh, I have to write by red color so that you won't get confused. It is N15 and this is also N15. They are going to get separated now. So now
Yeah.
the new strand is coming. So this new strand will be of N14 or N15.
14. 14.
Very good. So this new strand is coming. This is
Oh.
how this is how your DNA is getting modified day by day or
Oh.
in the new generation. This DNA is
Oh.
also N14.
Mm.
So this is also N14 and this is also
I see.
N14. So now just tell me, this DNA contain how much percentage of parental character?
50%?
Yes, and 50% is new. Yeah.
Oh, yeah, yeah, yeah.
If I see the older DNA, not the parental one, but the older DNA,
Okay.
this DNA also contain how much parental one?
The red, yeah, 50%.
Now, okay, then the again I am putting it into the same medium, like they are in the same medium again. We are not shifting it. So now, of course, all these are going to get separated.
Mm-hmm.
So now, how many DNA will get formed? Can you tell me? How many DNA will get formed?
4?
Yes, of course. So now all are separated, right? So if the medium is same and 14, so new DNA will be of.
And 14 as well?
So it means the new DNA is coming is N14. So N14 is something like this, right? Now
Oh,
all
yeah.
N14 are coming.
Oh, that's where. Okay, now it's 75% is
Yes.
the my character or like new character, whatever.
So now just tell me, this complete DNA contain
Is
how this
100%.
DNA contain how much character of the parental one?
None. 0%.
Zero. It means some new DNA is also there in your body.
Variation, yeah.
Okay, this contain how much?
50%.
This,
100.
eh? 100.
Oh no, zero. Okay, sorry,
And
zero.
this 100 is 100 can't be there.
No,
100%.
I
If
thought you meant a
okay,
new
if
character.
whenever you say 100% of parental, so it means it should be exact copy of parent, which is not possible.
I meant 100% new
100
character.
can be there only in the case of unisexual amoeba, something like this.
Mm.
So you are getting two DNA are completely new and
Yeah.
two DNA are completely hybrid, like 50-50%. So two are hybrid and two are completely new. So this is
Yeah.
known as the semi-conservative nature. Semi because it contains something na
Yeah.
50% conservative. So that's and two are completely new. So two hybrid and two new can we say it is 50-50%?
Yeah.
How many hybrid are getting?
Two.
Two new,
Two.
two, one by one. So 1:1. So it is 50% na. That's why we are calling it as semi-conservative in nature. Getting that?
Mm. And this takes place in humans, right?
Yeah, it is basically for all
Okay.
the animals. Okay. So this is how,
Mm.
this experiment, if I ask you, can you tell me the name of the scientist who gave like DNA is a semi-conservative in nature?
Matthew Madison Franklin Stanley.
Madison, no, Matthew Messelson and Stahl.
Oh, okay.
Say Messelson and Stahl.
Messelson and Stahl. Okay, then I have to rewrite my.
Yes. So this thing happens like 20 minutes and this is also like 20 minutes. So total it took 40 minutes. Okay.
And Stahl?
See, this is N15 and N14 also. So if in a test tube, if I say that test tube, so it is very heavier one. So it is you will find this is a bottom, medium, or at the top.
Huh? Say it again?
This contain N15, N15, and this
Yeah.
contain N14. So N14 is heavier or
Oh,
N15?
you cut out. Sorry.
N15 is heavier or N14?
And 15.
So 15 heavier, you will see at the downward.
Bottom. Bottom,
Okay.
yeah.
But now if I draw here, like test tube for the both, like both are same, so I can
Mm-hmm.
make only one. So you will find this at top, medium, or the bottom.
What? The blue one?
Both are same.
Oh, okay. Medium, I guess.
Yes, because one is heavier, one is lighter. So of course medium,
Oh.
medium.
And the fully blue one at the top, because it's the lightest.
Yes. So for
Uh,
this, so it is at the top because both are 14, right? This is also at
30
the
by
top.
928.
Yes. So it is basically for the 14 one, 14-15 medium, and 15-15 bottom. Got it?
Yes.
So the DNA extracted from the cell after one generation of transfer from the N15 to the after 20 minutes from the like to the N14. So we are going to get two are intermediate and two are completely new, right? Two are
Mm-hmm.
hybrid type and two are completely new, and this phenomena is called the semi-conservative in nature, which is given by the Matthew Michelson and this Hall. Got it, everybody?
Mm-hmm.
So this experiment was also conducted by the Taylor, scientist named Taylor, with the help of the radioactive thymidine. And finally, he also proved that DNA is a semi-conservative in nature. So if I ask you,
Mm-hmm.
Vishnu, what do you mean by the semi-conservative in nature, then what will you say?
Oh, the DNA has both parental, and its own variation.
What? Can we say whenever a DNA is going to
Oh,
replicate, so it retain only half of the character?
yeah. So like there's like two hybrid and two complete new character.
Yeah, so this is known as
One
the
ratio one.
hmm. So they're known as semi-conservative. It means half of the character remain conservative.
Yeah.
Conservation of half of the character. Okay, so it is done by the Matthew Michelson and this Stoll. You will remember that, yes or no, everybody?
Oh yeah.
Okay. So actually there is a diagram as I already showed you. Like if I show you by the fork-like structure, like there is a DNA. So of course, if I show you something like this, not the helical structure. So firstly, DNA will try to like separate it with the help of which enzyme? Which
DNA
enzyme?
helicase.
Very good, very right. Okay, now because of the DNA polymerase, new DNA are coming, right?
Yeah.
And it is also going like this, like polymerase is coming like this. But here, polymerase has to go like this. So sometime what happen? It is going to cut here. It is going to be like this at this side, not at this side, but at the bottom side. Why? Because polymerase is going
Mm.
like this, and it is going like this. So direction is opposite. So it is not so not so continuous. It is discontinuous actually. So if you want it to be continuous, so what we have to add? I told you these strands can be joined by which structure? Tell me.
DNA polymerase.
No, polymerase is basically doing the formation of DNA.
Ligase.
Yes, yes, yes. So DNA helicase is
Separation.
very good. And if I say
Ligase rejoin.
DNA polymerase, formation of new DNA, DNA without DNA polymerase, can you form one DNA to two DNA? And DNA ligase?
The DNA ligase is used to connect the two DNAs
Yeah.
together.
So this is known as the
And
lagging strand. Why? Because it is not so continuous. Lagging strand. And this is known as the leading strand. Leading strand. And I already told you because of the bonding, remember? If this is 5 prime, so this is 3
three
prime.
prime. Three
If this
prime.
is this is 3 prime, this is,
That's five prime.
so if the new DNA is coming, so this is 5, so this side is 3 prime,
Three.
and
Yeah.
this side
Mm-hmm.
5. If this side
And
5,
three.
this is 3. This is
What
3,
are they
this
called
is,
again?
this is
That
new.
that's the number of carbon. That's the number of carbon. Three
Yes,
five, right?
bonding.
Bonding.
Okay.
Cause I remember because the
5
nitrogen
and
is
3.
bonds.
Yes. Nucleotide structure.
Yeah, cause
One
the
nucleotide.
nitrogen bonds were inside, and then everything else was outside.
Yep. Okay. So
Mm-hmm.
this is how DNA replication taking place. So just if I say new DNA is coming from the pre-existing DNA. So this is
Mm-hmm.
right statement or wrong statement?
Say it again.
New DNAs are coming actually from the pre-existing DNA, already formed DNA.
That's false.
So new DNA is originally coming as a new. It
Oh no,
doesn't
it comes
have,
from parent.
yeah, I showed you without old
Semi-conservative.
these two DNA.
Hmm.
If I say that these two DNA are coming randomly as a new, it doesn't have any relation with this old DNA. Is it
I
right?
wish it did, but no, it's false.
Obviously. So it is going and going and going. That's why we say our character transfer from the first generation to next generation. Okay, and you are going to show same character which your parents have. Got it now?
Yes.
Yes. Okay. Okay, got it. Okay, so we
Mm-hmm.
have completed this portion. Okay, now in the next class we are going to start the like this. What? Wait a second.
Translation,
Oh,
transcription,
my
and stuff.
drawing is not so bad. I know that. Thank you.
What? No, I like it. I
Nothing,
like it. It's
nothing,
good.
nothing.
It's good.
Okay, DNA to mRNA. Okay, in the next class we are going to I like my self appraisal. Okay, so transcription and translation we are going to discuss in the next class. Okay,
Mm-hmm.
Vishnu, do you have any doubts? So you can ask as you were saying something for your school and all.